Good morning or afternoon, everyone. My name is Charles Godinez. I'm a rising junior from Barstow Community College. This summer, I had the opportunity to receive training, mentorship, and conduct a research project at the Laurent Lab at the University of California, San Diego. The title of my presentation is Antibody Selection for Geomic Segmentation of Cell Subpopulations in the Fallopian Tube Epithelial Mucosa. So the Laurent Lab is a translational lab that focuses its research on female reproductive organs to improve pregnancy outcomes. The lab works towards identifying biomarkers for diseases such as preeclampsia, placenta accreta, and preterm birth. With respect to HuBMAP, we're the Pregnant Female Reproductive Tissue Mapping Center, and our group's role is to create multimodal atlases for the placenta, fallopian tube, and uterine endomyometrium using healthy tissues. My project centers on the fallopian tube, which is an organ in the female reproductive system that serves as the channel for the egg cell, and notably where the sperm and egg meet and fertilization occurs. For this study, we analyzed the ampulla and fimbriated regions. So most of the transcriptomics data from the fallopian tube comes from bulk RNA sequencing and single-cell RNA sequencing. I have this fruit illustration that can help aid in explaining bulk and single-cell sequencing, as well as introduce spatial transcriptomics, which we utilize for this project. You can think of the fruits as individual cell types, each with their own unique flavor. With bulk sequencing, gene expression can be read out from the tissue, but does not provide cellular context. In other words, you can taste the flavor, but you don't know which fruit produces that flavor. With single-cell RNA sequencing, we do receive cellular context. However, the data lacks spatial information on where the gene expression originates from the tissue. Over on the right, we have a visual analogy of what spatial transcriptomics data provides. If we consider each different fruit a unique cell type, and we consider the cake as the tissue, we can pinpoint where on the tissue the expression is coming from and understand how gene expression can vary from different regions throughout the tissue. The aim of this research is to perform spatial transcriptomics on the fallopian tube, focusing on the epithelial mucosa. So the reason there's so much interest in the epithelial mucosa is that recent research suggests that ovarian cancer originates within this region of the fallopian tube. Since spatial transcriptomics of the epithelial mucosa is lacking, as I mentioned, most of the current data is from bulk and single-cell RNA sequencing, further research is certainly warranted. We utilize the Geomics whole transcriptome assay for this analysis. And so to provide an overall view of how we processed our sample, on the left, you can see an illustration of our tissue embedded into a formalin block. These are known as FFPE blocks. FFP stands for formalin fixed paraffin embedded. We use a microtome to cut thin 5-micron thick slices and mount the slices of sample onto slides, which we then analyze using the instrument shown at the bottom called the Geomics Digital Spatial Profiler, produced by a company called NanoString. And here's an overview of the entire assay workflow. And the key to this technology are the RNA probes. RNA probes are oligo tags connected to complementary sequences for the target RNA, and they're connected via a photocleavable linker. These RNA probes are allowed to hybridize on the tissue overnight. Then the next day, the user would stain the tissue with a nuclear stain to visualize nuclei and up to 3 fluorescently labeled antibodies, also called morphological markers, that will assist with the selection of regions of interest that the user wishes to profile. The region of interest, which we refer to as an ROI, is an area on the tissue selected by the user for spatial profiling. Using the fluorescent markers, which should be specific to a certain cell type, one can split up an ROI into several constituent AOIs, AOI standing for area of illumination, and this is called segmentation. Once the user selects an ROI and performs segmentation, the instrument shines UV light on the specific areas chosen, releasing the oligos and collecting them in a 96-well plate. Each collection refers to an individual AOI. Therefore, each well of the 96-well plate corresponds to a specific area on the slide. The next step is to prepare libraries from the individual AOI and sequence the libraries to determine gene expression in these specific areas. Because segmentation relies on morphological markers, antibody selection is an important aspect of experimental design. So on the left, we have a diagram with the various cell subtypes in the fallopian tube. We're interested in the cells in the epithelial layer, which are the secretory, ciliated, and PEG cells. Ciliated cells are characterized by these hair-like projections that help facilitate the movement of the egg through the fallopian tube. The secretory cell releases the type of fluid that also helps the egg travel through the fallopian tube. Then there are these basally located cells called PEG cells, which are the progenitor cells of the epithelial layer. Notably, these PEG cells only make up about 10% of the total cells in the epithelial layer and renew the secretory and ciliated cells when necessary. We're currently working to find an optimal antibody marker that specifically targets these PEG cells. So on the right, we have a chart showing the antibodies which we've selected and tested as unique markers for the ciliated and all epithelial cells in the fallopian tube mucosa. Starting with the first column, pancytokeratin targets all epithelial cells in the fallopian tube mucosa. In the following column, we have CAPS, which uniquely targets ciliated cells. And on the far right column, you'll notice we're still searching for the unique marker for the PEG cell, which will not only allow us to segment the PEG cell, but also the secretory cells by process of elimination. So what makes a good morphological marker? Well, some characteristics include it should be cell type specific to enable accurate segmentation. It should have a high signal to noise, and monoclonals are often better than polyclonals in this respect. It should be validated for immunohistochemistry, ideally on the fallopian tube. And preferably they should be conjugated to the appropriate fluorophore, although this is not a strict requirement because we can conjugate ourselves. And some resources we use to develop the list of antibodies we selected, we searched through the literature. We would cross-reference antibodies mentioned in the literature with IHC images of fallopian tube from the Human Protein Atlas, which allowed us to further boil down our list of candidates. And we referenced technical data sheets accompanying commercial antibodies as well. So in the following slides, I'll introduce the 4 antibodies we use to try and target the PEG cells, and following that, I'll share the results. So this is an immunohistochemical staining of a fallopian tube section on a slide stained with LGR5, where you see the brown staining that corresponds to LGR5 protein expression. The IHC looked reasonable, and the data from the literature describe LGR5 as being expressed in the progenitor population, so we decided it would be a good candidate. The second antibody we selected was SCOOP2. This antibody had promising IHC of the fallopian tube. So looking at the IHC image on the left, the brown stains correspond with SCOOP2 protein expression. And moreover, single cell data from the literature describes SCOOP2 as being primarily expressed in the progenitor population. If you look at the HuBMAP plot in the bottom center, the 2,5 and 2,2 regions are both cell clusters for progenitor cells. If you look at the HuBMAP plot to the bottom right, you can see SCOOP2 was primarily expressed in the progenitor population 2,5. The third antibody we selected was decorin. On the IHC image on the left, the brown stains correspond with basally located progenitor cells. And again, the HuBMAP plot here in the bottom right shows decorin is primarily expressed in cluster 2,2, which is a progenitor population. And here's the fourth antibody we chose, CD44. IHC shows basal staining consistent with the location of PEG cells, and this antibody was mainly chosen because it was strongly supported in the literature as a marker of stem cells in the fallopian tube. Okay, so now this is our high-resolution scan with the GeoMx from the GeoMx instrument. As you can see, it's a cross-section of the fallopian tube, and this is from the ampulla region. For this study, we're really only interested in the epithelial mucosa, which is in this region here. The empty space is referred to as the lumen, and this is where the egg and sperm travel through. Now, if I zoom in further, we can have a better view of the epithelial cells. Here I'm showing a merge image of all 4 channels: DNA, PanCK, CAPS, and LGR5. To provide some perspective on the zoomed in image, the lumen is the empty space. The outer row of cells encircling the perimeter is the epithelial layer, and underneath the epithelial layer is the lamina propria. So in the following slides, I'll show the scan of this area with each channel individually to show how each antibody and stain performs. So here we have the DNA channel, which stains all nuclei. And as you can see, the DNA stain looks good. This is the pan-CK channel. It also looks good. It's staining epithelial cells with high signal to noise. This is the CAPS channel, which stains all ciliated cells in the epithelial layer, which perform well as well. And here is the LGR5 channel. And fortunately, LGR5 only appeared to stain red blood cells in the lamina propria. Now, if you recall from the IHC image we showed earlier on the left, we expected LGR5 to stain for the PEG cell population, but unfortunately, the antibody did not perform as expected. And here's the geomic scan for SCOOP2, and unfortunately, we did not receive the results we expected. SCOOP2 also appears to just stain red blood cells in the lamina propria. And here's the geomic scan for Decarin, and it appears that this antibody appeared to stain the lamina propria, which is not what we'd expected per the IHC of the fallopian tube. And CD44 seemed to stain all epithelial cells. It does appear that too much antibody was used in this case, so we may want to dial back on the concentration if we decide to use this antibody again. So what are the next steps? Well, technically the run was successful. We were able to stain all cell subtypes in the fallopian tube epithelial mucosa using pan-CK, and we were able to successfully stain all ciliated cells using CAPS. But unfortunately, the antibodies we selected to profile PEG cells did not behave as expected. So naturally, we asked, why are the antibodies behaving differently in the geomic scans and in the IHC? Well, we identified a few potential areas to look at and troubleshoot. First, conjugation. We conjugated using a commercial kit, so it should have worked. In fact, CAPS was conjugated using the same protocol, and that worked really well. But we can always search for antibodies that are already conjugated. We found the IHC images on the Human Protein Atlas very encouraging, but these antibodies can only be purchased unconjugated. We may consider purchasing antibodies that are pre-conjugated from an alternate supplier. Next, we could look at the concentrations that were used. Generally, we need to use higher concentrations than suggested for IHC because the geomics protocol includes a proteinase K step, which results in the loss of epitope sites. Perhaps we need to use a higher concentration for some of these antibodies to compensate for the loss of epitope sites. Another area to review is whether the antibodies are polyclonal or monoclonal. And here's a cute cartoon to drive our point across. Two of the antibodies we selected were polyclonal. Perhaps monoclonal versions may work better. Lastly, we may decide to use new antibody candidates altogether. We may find better evidence that supports the use of an antibody that we did not use in the experiment and try that. So as a final note, it's worth continuing forward to find a marker for PEG cells, because if we're successful, we'll be able to profile all cell subtypes within the fallopian tube's epithelial mucosa. We can then utilize spatial transcriptomics to understand how gene expression varies among these cell subtypes across the region. Moreover, it's already known that there are more progenitor cells towards the fibrinated end of the fallopian tube. And this has important implications for understanding the mechanism of ovarian cancer initiation. Also, with spatial transcriptomics data, we can answer additional questions. For example, we can learn how gene expression may change as you move closer or further away from the lumen. Therefore, performing spatial transcriptomics in this region is important because it could lead us to deepen our understanding of the fallopian tube biology and how dysregulation can lead to disease. And with that, I'd like to finish just some quick acknowledgements. First and foremost, just want to thank the NIH for providing me with funding for this life-changing opportunity. NIH coordinators, thank you, Zarina. Thank you, AJ. Thank you, Megan. Thank you, Maya. I'd like to express my gratitude to my PI, Dr. Laurent, for granting me the opportunity to work in her lab this summer, which has been an absolutely transformative experience. I'd like to thank my mentor, Dr. Lindsay Hewitt. I'd like to thank my professors and administration at Barstow Community College. Shout out Dr. Nalbandian. And finally, I believe the adage, it takes a village holds true. And I'd like to thank the entire Laurent Lab team who are part of my success this summer, one way or another. This is just an image of our 4th of July party, which was a lot of fun. All right. So